Thanks to the organizers for the invitation to speak. My lab runs something called the Bioanalytic Resource for Plant Biology, and that's at bar.utoronto.ca, and that allows easy exploration of large biological data sets for hypothesis generation in plant biology. We've got lots of expression data sets, but also subcellular localization, protein-protein interaction, protein structures, lots of polymorphisms. We get about 3 million page views per month. So here's a concept drawing from a 2012 Genome Canada grant application where we wanted to build a zoomable user interface to really move between different layers of biological data from the kilometer scale, natural variation, through the centimeter scale, tissue level expression, all the way down to the nanometer scale, protein structure, DNA sequences. And you can see from that concept drawing that it's actually quite similar to the Plant Cell Atlas visualization published in 2019 by Sue and Dave and Ken. As part of this Genome Canada grant application, we developed something called ePlant, which does allow you to explore data from the kilometer to the nanometer scales in sort of a hierarchical manner. And you start by entering a gene identifier, and there are a couple of metadata levels, such as getting the annotation, the DNA sequence, the coding sequence, that kind of thing. And then we can zoom down to the first data level, which is at the kilometer scale, looking at variation in expression of a particular gene, a DOF gene here. We see some variation based on the coloring of these little plants here, and we can zoom down to find out what that ecotype is that shows that variation in expression. We can overlay climate data to start to think about the linkage between external climate and variations in gene expression. At the centimeter scale, we can see that this gene, which is known to be involved in phloem development, is strongly expressed in the roots and also in some of the apical meristem tissues. Again, zooming, we can also see the expression levels in an RNA-seq atlas, very similar, strong expression in the roots here. And we can zoom down to the millimeter scale and look at cell type specific data sets where we can see very strong expression in these, the protophlome tissues and the metaphlome tissues, as would be expected for a gene that's involved in phloem development. Then further zooming down, we've just added a single cell RNA-seq atlas from John Schiefelbein's lab, where you can again see strong expression in phloem cells here in this t-SNE plot from his lab. Going further down the hierarchy, we're now at the sub-millimeter level, looking at the localization of the gene product. It's strongly localized in the nucleus, found to be localized in the nucleus, and that's from SUBA data from Josh Hazelwood's lab. And given the fact that this is a transcription factor, you'd expect it to be localized in the nucleus. Of course, if you didn't know what the gene product did and you saw that kind of localization, you might suggest to you that that is involved in something related to transcription. Going down to the micrometer level, we can see where this gene is localized, which arm of chromosome 5. Further to the micrometer level, we can see the protein-protein interactors and also the protein-DNA interactors organized here by chromosome. The green lines denote that these are actually, there's some experimentally documented protein-DNA interactions. The dashed lines mean that those are all predicted. And then at the nanometer level, we can get a view of the predicted protein structure along with variation. So we've tried to integrate across scales, showing that there is considerable variation at this position, which actually lies close to this DOF domain. And then we can find out what variants have that, what ecotypes have that variant in them. And of course, we can zoom down also to the level of the DNA sequence using a JBrowse instance that Tara is now managing. So we can also move across species quite nicely, and we've got a navigator that allows you to do that. And then what I was thinking just for the last few seconds to talk about, okay, we've got this collection of parts, and if we've got that as an electronic circuit, we're not so much interested in that. It's important to document that, but we do want to move to thinking about things as a systems response, what kind of response an organism or plant gives. That's what we care about when we turn on our iPhone. We want to listen to the music. We don't really care about the wiring diagram. And I was thinking that we might consider something like OpenWorm, where you're allowed to browse between levels in a Google Earth-like manner. Here's the tissue view for the OpenWorm project. But the nice thing about OpenWorm is that they've integrated through a Kickstarter project something called WormSim, which allows you to run simulations and change parameters and see, say in this case, a worm wiggling through across your computer screen. All right, that's it for me. Thanks again.